Okay. Okay, hi everyone. Welcome back. Today we're going to move on into the vascular plants and talk about, let me share the screen, the lycopods. So let me make this big. Okay, good. And let me make this a little bigger too. Okay, great. Yeah, so today we're going to talk about the lycopods. Last time just another reminder, I kind of said this a lot last class, but in case the message didn't get through, just make sure that before you go to lab, you need to be prepared, right? So the lab materials are on Canvas a day or two before you go to lab, right? Friday even. Download the lab from Canvas, read through the lab, understand what the learning objectives are, and it's strongly recommended that you define the terms before coming to lab. And you need to bring your own copy to lab. Please do these things. Please show up prepared. But if you guys keep coming to lab unprepared, then Dr. Fonseca and I will make some adjustments and make things be a little bit more strict. So I don't want to do that because it's more work for me and her, and I want to treat you guys like adults. So just show up to lab prepared, please. Okay. So enough about that. Last time we went through the introduction of the plant body, so we looked at what steps were involved in the evolution of vascular plants from the non-vascular plants. We talked about some anatomy, right? Cells, tissues, things like that, as well as external anatomy like shoot system, root system, things like that. And then we talked about some phyla in these early vascular plants, right? So we talked a little bit about lycopods and horsetails briefly as kind of like a generalized. Today we're actually gonna talk in more detail about lycopods, right? So their diversity, the morphology of the lycopods, some of their evolutionary steps, and then a new concept that we need to add into our idea of alternation of generations, which is these terms homospore and heterospore, which we'll talk about later in today's class. All right. So I like to get oriented of where we are, right? So we've moved up into the vascular plants of the plant kingdom, right? And we're starting to talk about these first plants here, which are the ferns, lycopods, and horsetails, which are vascular plants that shed spores. So still no seeds are involved yet. So the phylum name for the lycopods is Lycopodiophyta. So it's a mouthful, but with practice, you'll be able to say it no problem. So here I show you how to pronounce it, right? Lycopodiophyta is the way. Myself and others call these plants the lycopods. Sometimes you may see in other resources lycophytes, same thing. This is pictures of some of these guys. So on the top here, we have lycopods that are known as Lycopodium, right? Lycopodium. And on the bottom, we have lycopods here that are known as Celgenella. So you can see it down there, Celgenella, right? Don't get confused. So both Lycopodium and Celgenella are lycopods, even though Lycopodium sounds like lycopod, right? The Celgenella is also a lycopod. Okay. The word lycopod, I always like to give you guys this stuff, comes from this Greek word lukos, which refers to wolf, and then pous, which refers to foot. So whoever was describing, let's use black. You know, somebody came across this plant a long time ago, or maybe even this plant over here, and thought to themselves, this plant looks like a wolf's foot. I mean, sure. I I guess so. We could use a little creative imagination, right? Like maybe. So lycopods, oh, my head's, I think, oh no, it's not. Yeah, maybe we're blocking the word plants, but so lycopods are the oldest surviving phylum of vascular plants. So what does that mean? We talked about some of these fossil plants in the last class when we were discussing the evolution of vasculature. Lycopods would have been around that same time, that around 400 million years ago, and they're still here, which makes them the oldest surviving phylum of vascular plants on Earth, to our knowledge. Wolffoot, I mean, but yeah, so they arose about 400 million years ago, very widespread in the Carboniferous period, which we'll talk about a little bit more at the end of this class. I don't know about the wolf's foot thing, but you know, if we were to like think about a wolf's foot, I guess you could use a stretch of the imagination here. Like, yeah, okay, fine. It looks like a wolf's foot. There's a lot of species. So there's about 1,200 known species. There's also many extinct species. So these are around 1,200 that are still around, but there are also many extinct lycopod species. And of course, because we're in the vascular plants, we know that the sporophyte generation is the dominant generation. We should know that already by now, right? Okay, wolffoot. So Carboniferous period, I guess we'll talk about it now a little bit instead of later in the class. So what was that? Well, so you have to think back in the geological history of the Earth. You may not be aware of this, maybe hopefully you are, but there's like tectonic plates on Earth that move the land masses around, right? When we have earthquakes, for example, that's an example of those tectonic plates moving or crashing into each other. And so where the continents are right now is not where the continents have always been, and are not where the continents will always be, right? Like they're all moving. In fact, for example, right now, the continent that has India on it is colliding into the continent that has the rest of Asia. And so that is what's causing the Himalayas to continue to grow and be taller and taller as those plates collide. Anyways, so if we were to go back around 360 million years ago and we were to look at the Earth, this is where the continents would have been. So just to show you some of maybe familiar players here. So like here's North America, kind of right at the equator. Africa is like way down, smushed with Antarctica, and India down near the South Pole. South America, not too far away from that. You know, Europe, Siberia, etc., China. So way different than it looks like now. And because of this, you could see like this all the continents are kind of squished together, which was a characteristic around this time in the Carboniferous. And the reason why that's important, the two major subcontinents, you have this Gondwana, which is like, oh, sorry, wrong one. Gondwana, which is like this mass, and then you have another subcontinent, which is Euramerica. And so in Gondwana, it was super cold, right? You're by the South Pole. And so you had a lot of ice and a lot of land that's basically covered in ice. And so this allowed the lots of seawater would have been kind of mixed with that ice, incorporated in that ice. And so globally, the sea level was very low. And this landmass here, Euramerica, is at the equator. There was also a lot of, you can see this lighter colored blue. It's kind of showing you kind of like really shallow seas all around that continent. And remember, this is at the equator, so it would be super tropical and temperate, tropical there, and subtropical. So a whole different vibe than what's going on now. Because of this, right? So it's like this lighter blue color I'm trying to show you. See this like lighter blue color that's surrounding that area. So those areas allowed there to be a lot of shallow seas and swamps. And that's kind of like a picture here of what it might have looked like. This is a reproduction, obviously. There's tropical conditions year round because you're at the equator. And the plants that grew in this swampy environment are much of which we use in our fossil fuels now. Hence, you can think of why it's like the carboniferous, right? Coal is made out of carbon. And so Carboniferous period, we get a lot of our coal originates from organisms that live during the Carboniferous period. If you want to learn more about that, you can look in the textbook on page 400, talk about these plants and the coal that we get from them. There were 4 types of plants that dominated these Carboniferous swamps. You had the Lycopods, you had the horsetails, you had ferns, and you have gymnosperms. So just to give you some context. In fact, as we'll learn later in this lecture today, like this big plant right here in the middle is actually an extinct type of lycopod. Okay. Oh, great. So there you go. So that the one I just circled is that Lepidodendron, which is this extinct lycopod trees. They were the most common plant in the end part of the Carboniferous and contributed a lot to modern day coal. But we will talk about this plant in particular a little later in the lecture. Okay. So just before we move on, some general characteristics of the phylum Lycopodiophyta. So there's the 2 largest genera are Lycopodium and Celgenella, which I've already mentioned. And Lycopodium that has about 400 species. Celgenella has a little bit more, almost double, probably a lot undiscovered so far too, because these do live still in a lot of tropical environments that there may be additional species. Lycopodium, there's also some debate. So you might be surprised to learn that like when a plant or when somebody names a plant genus or even a plant phylum and groups plants in that genus and in that phylum, that can change. As we learn more about the different plants, we start to figure out, oh, these are not as closely related as we thought. And so right now, Lycopodium as a genus is actually being divided into many genera. So there may actually be 15 genera inside of Lycopodium, but for the purposes of this class, we'll go with the old way and just stick with Lycopodium as being a single genus. As I was saying, this is how many species we know about. A lot of these species are tropical and might live in very like dense forests. So there's probably additional ones. The sporophyte plants can live just on the land, or they can be epiphytes, which is a term we learned already, meaning they can grow on other plants. So here on the left is an example of Lycopodium growing terrestrially, like just on the ground. And on the right is an example of Lycopodium growing as an epiphyte on other plants. I actually took a picture just like this many, many, many years ago when I was doing field work in Massachusetts for Tufts University. And I came across this in the forest. And I didn't know what lycopods were at the time. And I thought somebody had like brought their Christmas tree out into the forest and buried it underneath the ground. I couldn't figure out for the life of me what was going on. It wasn't until later that I learned that these were lycopods, but that's kind of what it looks like if you're walking in and don't know what they are. So those were examples of Lycopodium growing in the ground or on other plants. Same with Celgenella shown here. There's the sporophytes on the left growing in the ground, and on the right You can see how many of them. This is kind of cool. Like this is one big tree branch, like of a big tropical tree. You can see it's just like covered in Celgenella. Notice too, because this is a good opportunity to point out this, I'll use pink, I guess. Notice already what, like grow split, right? But over here, just grow, right? Ooh, something is happening there. All right, we'll see later what. General characteristics of the sporophyte. So the sporophyte generation is dominant. We're in the vascular plants, so we should already know that. In addition to being vascular, sporophyte plants have true roots, true stems, and leaves. And this is something, a new topic, but new concept, but something that we'll see many times going forward. So far in the class, here, let me get rid of this so you're not reading. So far in the class, when we've talked about a spore, right, it's just been one type of spore, right? In the true mosses, peat mosses, liverworts, hornworts, they are just making, we just say a spore, right? They make it, the sporophyte plants make a spores. Because they all make the same type of spore. Like each true moss species, all the spores that it's making are identical. But as we move forward, we're gonna see that that's not always the case. So for the Lycopodium is a good place to first talk about this. So some species, so I should say some sporophyte plants, right, of species that have on them sporangia and make spores, sometimes the spores are all the same size. And if that's the case, we call those spores homosporous. I think I talked about this in last class, but homo means same and sporus refers to spore. And that's how it is in Lycopodium. Lycopodium is like the other plants we've already covered in this class. They only make one type of spore. However, if you look at other plants, including Celgenella, plants make two different types of spores. Two different types of spores. We'll talk about them in a little bit. But think one way to organize this information is if a sporophyte plant produces homosporous spores, or one type of spore, right? That are all the same size. Those spores, when they germinate and grow into gametophyte plants, will produce gametophytes that are bisexual, meaning they will produce gametophytes that have both sex organs on one plant individual. However, if a sporophyte plant makes spores that are different sizes, two types of spores, two different size spores, each of the spore type will grow to become a gametophyte plant. One type will grow into a female gametophyte plant. One type will grow into a male gametophyte plant. So this is a good time for you to start thinking about those terms that you can use to describe whether a plant has both reproductive sex organs on it or one or the other, right? So what you can pause the recording and think to yourself, right, if a gametophyte plant has both sex organs on it, it is, right? Not even going to tell you. Oh, see? So you figure it out. And you could write it in the discussion if you want. That could be one thing you write. Okay, so that's a good way to think about things. What else? I pointed out on the previous slide, right? You do have that equal dichotomy again showing up. So one way you can tell how old a lycopod, so either Lycopodium or Celgenella, but a lycopod, one way you can tell how old it is in evolutionary time is how complex its branching pattern is. So, for example, the most primitive type or the oldest type of lycopod. For example, this species of Lycopodium here has equal dichotomy, which is that primitive thing we looked at, the grow split, grow split. So if something just has grow split, it's older in terms of evolutionary time. However, later on, as the as Lycopodiophyta, the lycopods, as they adapted and evolved, you'll get some members having unequal dichotomy, which is more advanced. So what that means is you still might have like a grow and then a split, like this here is a split, but then it's kind of unequal, whereas like it splits this way here, and then this whole thing goes on to split, and then maybe this one stops, but like this one goes on, and just so it's not as equal as, say, like that. And that's more advanced. I should point out, I did say branching pattern a second ago, but technically these aren't branches, and we'll understand that more in another lecture, just to keep it straight. Okay, so this is Lycopodium, right? An example of a more primitive member that has only equal dichotomy, an example of a more advanced member in one that has unequal dichotomy. Celgenella can have both. It depends on which species you're looking at, but most of them actually have a combination of both equal and unequal dichotomy on a single individual. So a good one to look at that could be this one here at the bottom, because we could see more of the plant. But so like the equal part is easy to see, is somewhat easy to see. You could see like here, like for example, here you got the grow, and then this here splits. That I did not draw that well. But like I'm trying to highlight this one here, right? Where it, you know, it grew and then it split right there, right? So equal is a little easier to see. Unequal is a little more just not so uniform, right? So like if you think of this stem, you have a split here, and then as the stem grows, you have a split here, right? And then here, and then here. This is still growing up, right? Here. So that's unequal, right? That's not, this that I just drew is not the same as that, right? One is grow split, one is grow a little bit, something goes this way, grow some more, now something's going this way, a little different. Celgenella usually is both. And just to be clear, all of these pictures show different species of Celgenella. All right. These are some terms we saw last time. So we know that we're in vascular plants, so the stems are going to have those tissue layers: epidermis, which is the dermal tissue system; cortex, which is the ground tissue system; and then a protostele, which we learned last time, just refers to a very basic arrangement of the xylem and the phloem in the stem and the root. Other terms we learned last time, right? Microphyll, oh my gosh, cannot draw. Microphyll, so for microphyls, you mean small leaf, right? Phyll means leaf, micro refers to reduced, small size. They have a single vein. And the roots are adventitious. We covered that last time too. That's just kind of a refresher. This is another refresher, right? If you're talking about Lycopodium or Celgenella, right, or the Lycopods, Lycopodiophyta, where are the sporangia located on the sporophyte plant? They are located on leaves. I said this a bunch of times last time too. Sporangia are located on leaves, not on the tips of stems anymore, on leaves. Those leaves are microphylls, because that's all that lycopods have. They only have microphylls. If a microphyll has a sporangia on it, has a sporangium on it, we can call that microphyll a sporophyll because it's a fill or a leaf that has a sporangium on it. And last time we also talked about this, that the sporophylls themselves, sometimes they look like normal microphylls and are green and just born singly, which means just on their own. They're not arranged anyway. It's like, here's one, here's one, here's one. They're not like arranged. Being like that, looking like a microphyll, looking like a regular microphyll, and being born singly, that's the more primitive state. However, over evolutionary time, some species have their sporophylls organized into this structure called a strobolus, which we talked about last time, and that's the more advanced way. And like I said, sometimes the sporophylls are photosynthetic. I don't know, let's see, let me just look at what I do on the next slide, because it may be worth it to draw. Right. Right, okay, yeah, it would be worth it to draw. Okay, so let's just do this. So on the left here, I'm gonna draw a simple, like a more primitive lycopod. So here is the stem, and on the stem are microphylls. The microphylls are not arranged in any way. They're just born singly, right? Here they are, little microphylls all over this stem, not arranged in any way, born singly. And in the case of these, they are green, so they're photosynthetic. And I'll use white to draw sporangium, or I'll use white to draw multiple sporangia. So let's say like there's a sporangia on this one, there's a sporangia on this one, there's sporangia on this one, there's sporangia on this one. All of these leaves, which are microphylls, all of them. But to be more clear and correct, the ones that have sporangia on them are called sporophylls, right? So sporophylls are microphylls, in this case, microphylls that have sporangia on them. So that's an example of a more basic, primitive, right? More primitive, basic one. And on the left, I will draw a more advanced one using a different color green. So here we go. So, all right, this one, here is the Lycopod, here's the plant. Oops, I'm trying to draw. All right, so these horrible things down here, these are regular microphylls on this plant. And then up here, I'll draw, yeah, I'll draw like this. All right, so here you have another lycopod, right? And on the bottom is the regular microphylls, these guys down here, right? But in this case, the sporophylls, or the microphylls that have sporangia on them, right? The sporophylls are organized in a structure called a strobolus. So that this thing up here is a strobolus, and on it are a bunch of microphylls. And in this case, these microphylls that are in this strobolus are not green, not photosynthetic, right? Okay. Hopefully that helps. In addition, sometimes the strobolus can be elevated and by a structure called a peduncle, and other times it could be not. So if the lycopod looks like this, and the strobolus is just right attached to it on the top like that, we call that sessile, and that's the more primitive state. Sometimes though, and let's say there's like, you know, leaves on the microphylls on the thing. Sometimes though, the more advanced lycopods will have their strobolus elevated by a structure called a peduncle. And in that case, it would look like this. There would be this long stick thing holding the strobolus way up there, and there wouldn't be any like leaves really on that. Okay. Now that I drew those very good drawings, let's look at it on real. Right, this is just a reminder. We already know that the spore mother cells make haploid spores. We already know that. All right, talk about Lycopodium first. So these are some examples of Lycopodium. This first one, Lycopodium lucidulum. I think you'll see herbarium specimens of this in the lab. Notice the traits that I just went over, right? Notice the traits. This one has equal dichotomy, right? See? This one has sporophylls that are photosynthetic. Here they are. They're green. And they are not organized in a strobolus, right? They are born singly. And you can see these Lycopodium lucidulum in and around New Jersey. So that's one. Lycopodium anotinum is stiff club moss. Also, you can see this in New Jersey, but look at how this one's a little bit different than one we just looked at, right? First, it has unequal dichotomy, right? So grow and then grow, but not necessarily split equal all the time, right? So unequal dichotomy. And the sporophylls, in this case, my head might be blocking it. But here, on this here, this is a strobolus, this is a strobolus, there's two strobili right there, there's a big strobolus right here. On this down here, you can see the strobili at the tops. Not elevated by anything, just attached, right? No peduncle, see, no peduncle. So arranged in a strobolus. The sporophylls are arranged in a strobolus. Next one is Lycopodium flabelliforme, which is fun to say, flabelliforme. Fan club moss. This one is even a little bit more advanced because you have equal and unequal dichotomy. You still have strobili, but check this one out. Not only does this one have many strobili grouped together, right? Look how many strobili are here. But you're also elevated by peduncle. So there's that peduncle thing I was telling you about. This one has multiple strobili elevated by a single peduncle. Other species have only one strobolus per peduncle. All right. So those are some examples of sporophyte plants of Lycopodium. Now let's look at gametophyte plants of Lycopodium. So gametophyte plants of Lycopodium can come in two different shapes. Remember that I said before, my head might be blocking it. Lycopodium sporophytes are homosporous, so they can produce only one type of spore. And when you're homosporous, just like we saw in the true mosses, peat mosses, liverworts, and hornworts, if you, let's say this is a spore that lands on dirt, when the gametophyte plant germinates, when the spore turns into the gametophyte plant, right, via mitosis, like say it's a phalloid shaped one, obviously the gametophyte plant is outside, it grows outside of the spore, right? The spore germinated and the gametophyte plant is dominant. And so when it grows, it's going to be much bigger. Like let's say the spore was that big, right? It's going to grow outside the spore wall because the spore has a little wall. When the gametophyte plant grows outside of the spore wall, we call it exosporic. Exo means outside of. So the gametophyte is outside of the spore, exosporic. But Lycopodium has two types, so they only make one type of spore. I want to be very clear about that. Lycopodium is homosporous, but depending on what species you're looking at, when those spores germinate, Some gametophyte plants, for some species, look more similar to what we're used to, right? Green blob. Other species of Lycopodium, when the spore germinates, the gametophyte plant almost looks like a little radish or a little carrot. And that's just species specific. It depends on what species you're talking about. But if you're a Lycopodium, you make one type of spore, and your spore, when it germinates and grows, the gametophyte plant is going to be outside of that spore wall. In some species, green blob, and other species looks like a carrot or a radish. The gametophyte plants, if they look like a green blob or if they look like a radish, they are very small. They are non-vascular, the gametophyte plants, non-vascular, and have those things we're familiar with, right? Rhizoids and things like that. Remember, I said if you have one type of spore and it germinates into gametophyte plant, now this is in the vascular plant. So if you have one type of spore in the vascular plants germinates into gametophyte plant, you'll have gametophyte plants that bear both sex organs or are monoecious. So this green blob would have both male and female sex organs on it, and this carrot would have both male and female sex organs on it. Okay. Right. This is what I was saying. Sometimes they look like green blob. Sometimes they look like not a green blob. The non-photosynthetic ones. Oh, this is, don't worry about this. This is just showing you. Yes. So the two different types. Green blob is photosynthetic. So it has to be exposed to the sun. So green blob type of gametophyte plant grows on the surface of the soil, whereas the non-photosynthetic type of gametophyte, the one that looks like a carrot, grows underground. So here, subterranean gametophyte means grows underground. And you might think, well, how does it live then? Because it's not being exposed to the sun and it's non-photosynthetic. So how does this carrot radish looking gametophyte plant actually live? And the way it gets help from special cells, these mycorrhizae cells, but also more importantly, this symbiotic relationship with fungus. So if you look here, this is your carrot-like or radish. What's a good color? Because there's a lot of colors on this slide. Yellow. So you can see here, this is your carrot or radish-looking gametophyte plant. This is it under the microscope slide, like a cross-section through a microscope slide. And what this is showing you or telling you on the slide is that the carrot-looking gametophyte plant has this symbiotic relationship with a fungus called mycorrhiza, right? Plural mycorrhizae. And some species of Lycopodium can't live without it. They have to, and those are the ones that live underground. The gametophytes live underground and are non-photosynthetic, like these, right? They will receive, so the mature plants will establish a relationship with this mycorrhizae fungi. And those mature plants will give the fungi sugar and receive water and nutrients from the fungi. But in addition to that, it serves to vector sugar to the subterranean gametophyte plant. Okay, I'm spending way too much time on this. But just so you know here, what I want you to know is that some species of Lycopodium have a symbiotic relationship with mycorrhizal fungi. That's what I want you to know. And just to hit it home, this is a cross section through gametophyte plant that's in the circle. And this layer here, this purple layer here, this purple layer here, those are actually fungi that have stained in that microscope slide. And if you were to zoom in close to those fungi, this is what they would look like. All right. Symbiotic relationship with fungi, mycorrhizal fungi. All right. Interestingly, this is something that's been going on for a really long time. So if we looked at some fossil specimens of Aglaophyton, remember, which is our kind of intermediate plant, right? They actually have this relationship with mycorrhizal fungi. So this is an actual, the picture on the right is showing you fossilized fungi inside of this plant fossil, which is pretty cool. Okay, now we're going to go through alternation of generations. Yep. Of the Lycopodium, and then we'll talk briefly about Celgenella and how the two types of Lycopods, Lycopodium and Celgenella, have slightly different little tweaks to the alternation of generations. So this is a diagram from your book. And so we'll go through, we just learned a lot of these terms. I'm going to change colors to orange. Okay, so look, here's our little carrot gametophyte plant, right? Not green, not photosynthetic, haploid, obviously, because it's gametophyte plant. This gametophyte plant came from a spore, came from one type of spore. So we know that this gametophyte plant grows outside of the spore wall, right? Grows and has male and female sex organs on it. Once the sperm and egg come together, it will grow into the new sporophyte plant. Here you can see the sporophyte plant emerging from where the egg was. Here's the roots of the sporophyte plant going downward. Talking about Lycopodium, right? So some of the terms that we've already covered, look, here's that horizontal stem, right? Rhizome. This species here has its sporophylls arranged in strobili. In addition to that, this species here has the strobili elevated by a peduncle. And since it's a Lycopodium, we know that it has microphylls all over it, right? Okay. If we were to zoom in to the top of one of these strobili, right, to look at just a single strobilus, you would see sporophylls, which are basically microphylls that have sporangia on them, right? So look, sporophyll. So this here is one sporophyll. This here is one sporophyll. This here is one sporophyll. Right over here might be easier to see. This is one sporophyll. This is one sporophyll. And on each sporophyll is a sporangium, right? So like here's one, etc. Right? If we were to zoom into one sporangium, it would be things we're used to seeing, right? Sporogenous tissue that's diploid, that's going to undergo meiosis to make spores, right? That part we're used to. So just note that the sporangia are on sporophylls. We saw this before, but this is a nice microscope slide to show you sporophyll bearing sporangia, right? Here's the sporophyll. Here's the sporangium coming out on top. Oops, too many boxes up here. Okay, right, call that. So this is where we left off, right? Meiosis is going to occur in the cells that will generate the spores, the sporogenous cells. Once meiosis occurs, the result is haploid spores. So this here is our sporangium. You can see there's all these things inside of it now. Those are the spores. Here's a spore that's leaving. Look, it's gonna fly away. And if we were to zoom in and look at that spore, this is what we would see, right? This is the spore. Notice that this spore wall is like starting to rupture. And this thing coming out is the gametophyte plant, which eventually will grow and grow, but via mitosis and become a big, green blob in this case. All right, so there's our germinating spore. We know there's only one type of spore here, so that germinating spore is going to germinate and grow into gametophyte with both male and female sex organs on it. Here we go. Just to keep the same colors as before. Here is the spore wall again, right? This thing here is the spore wall. And that thing coming out is the gametophyte plant emerging, and look how it's growing out of the spore wall, right? If we were to look closer at that gametophyte plant, we would see the female sex organs, male sex organs. Eventually the sperm and egg will come together. This is just a note when you're looking at the slides later, right? Homosporous spore grows bisexual or monoecious gametophyte plants, right? And the gametophyte plants grow outside of the spore wall or ectosporic. So this is just, look at this this is a plant sperm, right? So mature antheridium is going to make sperm. Mature archegonium will have an egg in it. Eventually sperm is going to swim and fertilize the egg. And when that happens, you go to your 2N zygote, which is the beginning of the sporophyte generation. 2N zygote, we already know this. Been doing it millions of times now, right? 2N zygote is going to grow via mitosis into the young embryo, grow, and then back into that mature sporophyte plant, as we see on the left. Okay, that was the life cycle of Lycopodium. So now let's talk a little bit about Celgenella, and then we'll talk a little bit about the prehistoric lycopod that I mentioned at the top of the class, and then we'll be done. So I showed you example species of Lycopodium. These are some example species of Celgenella. So this one is Celgenella apoda, something you could also see in New Jersey and New York. And notice the unequal and equal dichotomy, because I said Celgenella tends to have that. And Celgenella has usually all the time strobili, but they're usually not as conspicuous. They're usually not as noticeable. So like, technically this is a strobilus up here. Technically this is a strobilus up here. And likewise, technically there's strobili here, but they're photosynthetic and they look kind of like regular microphyls. That's what I was telling you. So this is another one. This one's not found in New Jersey, New York area. This one is a desert species of Celgenella. This has equal and unequal dichotomy as well. It also has strobili that are toward the tip, but again, look a little bit more strobili. But this one's super cool because it's called the resurrection plant. And so this plant lives in the desert and doesn't have a lot of water there. And so a lot of the time the plant lives, it looks like this. And then once a year when the monsoon rain comes, or I think, is it called monsoon there? I'm not sure. But when the big rains come once a year, in a very short period of time, the plant goes from looking like this little dried up ball to looking like this. Hence why it's called resurrection plant. And I have a video that I can show you. So stop share and I'll grab this video of the resurrection plant. Oh, I already have it up. Okay, go back here. and share that. All right, you should be able to see the YouTube video now. So this is the plant. We put it in water. And look, you can see the time elapsing there at the bottom. This is happening over the course of minutes, right? Like several minutes. But yeah. And then as soon as this happens, it'll release the spores. The spores will germinate, and the sperm and egg will fuse. Well, the spores will germinate into gametophyte plants that make the egg and sperm. Then the egg and sperm fuse. You know it. Whoa. Hold on. Sorry, YouTube is trying to show me more stuff. Okay. Let's go back. Yeah, so resurrection plant, pretty cool. Yeah, I used to have them. I used to bring them to class. You can find them online for sale. Okay. Yep. So, right, really trying to hit this home about this new term, homosporous versus heterosporous, right? So Lycopodium is homosporous. Lycopodium makes one type of spore, right? But Celgenella sporophytes are heterosporous. They make two types of spores, And this is a new concept. And those spores are called microspores and megaspores. So you might remember earlier when I was introducing microphyll, and I said that for the purposes of microphyll, micro means small and phyll means leaf. So small leaf with one vein. But aside from microphyll, all the rest of the class, when we see the term micro, it refers to male. And when we see the term mega, it refers to female. So when you have microspores and megaspores, there's a few things that are different than we're used to. Number one is that when you have microspores, the gametophyte plants actually grow inside of the spore walls, which is something we haven't come across yet. If a gametophyte plant grows inside of a spore wall, it's called endosporic. So just to slow down a little bit here. Who can be homosporous or heterosporous? Sporophytes. Sporophytes are heterosporous, and then in Lycopodium, sporophytes are homosporous. It's the sporophytes that we can describe that way, right? But if a gametophyte plant grows inside of, like, who's endosporic and who is exosporic? The gametophytes. So gametophytes are either going to grow inside of the spore, is the case for these heterosporous, when there's two types of spores, or gametophytes grow outside of the spore, as we saw in Lycopodium. Just trying to make that clear. Sometimes students get confused and they want to say that gametophytes are heterosporous or homosporous. I'm just trying to make it very clear, like, it's the sporophytes that are either homo or heterosporous, and it's the gametophytes that are either endosporic or exosporic. Anyways, I'm getting distracted. Back up. Lycopodium sporophyte plants are homosporous. Celgenella sporophyte plants are heterosporous. They make two different types of spores. What kind of spores do they make? They make microspores and they make megaspores. Microspores are going to grow the male gametophyte plants. Megaspores are going to grow female gametophyte plants. But when you have micro and megaspores, the gametophyte plants stay inside of the spore wall, or can be called endosporic, right? Germinate or grow inside the spore walls. Male gametophytes inside of microspores, female gametophytes inside of megaspores. So this is what I was saying before, and you can see this when you do the slides later by yourself, but yeah. So from now on, except for microphyll, from now on, if you see the term micro, you should think male. See the term mega, you should think female. But to be fair, The reason why microspores are called microspores, in addition, they have male components in them, meaning they're going to have male gametophytes in them eventually, but they're small. Like, look at the size of the microspores compared to a megaspore. Megaspore is much bigger. And this is something that you see a lot in nature, where the female structures are bigger than the male structures, because they have to hold a whole egg inside. Inside of each microspore will be one male gametophyte plant. Inside of each megaspore will be one female gametophyte plant. And even though it grows inside of the spore, it still has sex structures, but it's just everything's much reduced. So if you have two types of gametophyte plants, a male gametophyte plant and a female gametophyte plant, you're dioecious, right? Right? Two houses. Yeah, you might see this term megagametophyte in some of your book and stuff, but it's the same thing. Female gametophyte is fine. So female gametophyte grows inside of this megaspore. I'll use the same colors as before. So here's that megaspore wall. This whole thing is the megaspore wall. And here's the female gametophyte inside, growing inside. Now, it does poke out a little bit. It does kind of like with rhizoids, like it does poke out just a little bit. But the whole thing basically stays inside the spore wall. And on that female gametophyte plant will be sex organs or archegonia, right? If you look at the male gametophyte plant, it grows inside of the microspore wall. So this stuff on the outside is a microspore wall. On the inside, you have the male sex organs, but what's happened microspores are so small. Microspores are so small, you can't really fit that much in them. And so what has happened is the entire male gametophyte plant has now been reduced to one antheridium. So inside of a microspore is one antheridium, and that's it. It's a very big reduction in gametophyte size. To go think about it. Think back into the true mosses and peat mosses, where you're walking through the forest and you see all this green fluffy with your eye, right? And that's all gametophyte plant. Now we're talking about gametophyte plants that stay inside of a spore wall. It's so very small. The megaspore is a little bit bigger, so it can fit like a few archegonia. It even has rhizoids. So female gametophyte could be a little bit bigger because it grows in the megaspore, but male gametophyte plant is only one antheridium now because it has to fit inside of the microspore. Now we're going to go through Celgenella alternation of generations so that we can compare it to the Lycopodium that we just went through. This diagram comes from your book. So same players as before. I'm going to go a little quicker through this. So here's our young sporophyte plant emerging. Here's our mature sporophyte plant. All right, some of the things we learned today, strobilus. Mature sporophyte plant has real things, right? Real stem, real root, right? Real leaf. If we were to look at one of these strobilus more closely, right? And up close, because Celgenella makes two different types of spores, it has two different types of sporangia, each type making its own spore type, right? So if a sporangium makes microspores, we call it a microsporangium. If sporangium makes, if a sporangium makes megaspores, we call it a megasporangium. And if you look at them under the microscope slide, you can tell the difference. Look how big the megaspores are. Here we can see 3 inside of this megasporangium. We see 3 big, big megaspores, right? Look how small the microspores are, and look how many there are in there. So you can tell under the microscope whether you're looking at a microsporangium or a megasporangium because of that. If a leaf or a sporophyll has a microsporangium on it, you can call that leaf a microsporophyll. If the leaf has a megasporangium on it, call that leaf a megasporophyll. This is why it helps to build this terminology of phyll, micro, mega, all those things, because then when you get to this point, it becomes quicker to remember. So if we were to take one of those microspores, here's a microspore that is leaving the microsporangium, and then down here is a megaspore that is leaving the megasporangium, right? And look at them terminating. This is just to draw your attention to, these are the things I just said. Don't worry about ligule right now. Oh, but there it is. That little doodad is a ligule. I'm not worried about ligule right now. Oh gosh, I guess I am. What is a ligule? Ah, geez, I thought we were going to go on to the spores. Okay, briefly, a ligule is a thing. We don't really know what ligule does. Ligule shows up on some plant structures. In Celgenella, it is on the leaves. Don't worry about the adaxial thing right now. We don't know what it does. Maybe it secretes water, maybe it absorbs water, maybe it's for protection, maybe it's leftover, like a leftover thing, like a vestigial thing. We don't know. This is just more prefixes so that when you see these later, adaxial means on the top side of the leaf, adaxial side of the leaf. The way I remember top or bottom of leaf, and this will come into play later, but since it's here, ab means away from or off. But the way I remember it is abaxial is the bottom of the leaf, the B for bottom. I don't really care about this right now. We'll learn about it more later. Okay. So we ended up here, right, with microspore leaving the microsporangium, megaspore leaving megasporangium. We're talking about Celgenella here. Inside the microspore is going to eventually be a male gametophyte plant, which has been reduced to a single antheridium. So you can see its trajectory going this way. And here you go, single antheridium inside, eventually going to make sperm, right? Which will leave. Inside of that megaspore is the female gametophyte plant. She sticks out a little bit, but basically stays inside of the megaspore wall. On that female gametophyte plant are many archegonia that each have an egg, right? Eventually, sperm will swim, fertilize the egg. And just a reminder, because these are new things, remember the sporophyte plants of Celgenella are heterosporous. The gametophytes in Celgenella are endosporic, meaning that the gametophytes grow inside the spore wall. Because they grow inside the spore wall, and there's two types, we know that, you know, there's a male gametophyte and a female gametophyte. We know the gametophytes are dioecious. Okay. Eventually, egg and sperm fuse in fertilization, and you have your 2N zygote. which is going to grow via mitosis. It's going to grow where the egg was, so it'll grow out of the new sporophyte plant will grow out of the megaspore wall because that's where the egg was, right? The female gametophyte plant was in there with her egg, so that's where the new sporophyte plant will be. And that's where we started this whole cycle here. Okay. Oh, this is the real thing. So this here is a microscope. This is a drawing of a new sporophyte plant emerging from the female gametophyte plant inside the megaspore wall. And here you go. So the green thing, this is the new sporophyte plant. This is the shoot of the stem of the new sporophyte plant. Here are the first leaves of the new sporophyte plant. Here's the root of the new sporophyte plant. And then in here, this would have been the megaspore. The female gametophyte plant would have been inside of there with her egg, right? Okay. And this is where we started the whole alternation of generations. So it's another, I really want to hit this home about why it's such a big deal that now we have heterospory. This is a big deal in terms of the evolution of plants. So recall from the last lecture, lecture 5, recall that these are the important characteristics of the non-vascular plants and the important characteristics of the vascular plants. And notice on the non-vascular plants, they have this reduced sporophyte, right? But in the vascular plants, we see the reduction of the gametophyte plant, right? And so that's important. Why? Well, so let's take a step back and let's really think about this. These were the non-vascular plants that we learned so far, or that we learned in this class, right? We learned the true moss, we learned peat moss, liverworts, and the hornworts, right? And in all cases, in all of these pictures here, the green stuff is the gametophyte plant, right? Gametophyte plant is dominant in the non-vascular plants. So it's huge, right? It's huge, basically, compared to where we're talking about now. And in addition, for all those plants, in the non-vascular plants, all of them only make one type of spore. And the gametophyte plants are really big, right? You see them with your naked eye, so they grow outside of the spore, right? They're not going to stay inside the spore. Gametophyte dominance, we know, but notice also that the gametophyte plant still does, as we go through this order, we still see a reduction in gametophyte size, right? If you think about the moss, true moss, even sphagnum moss, all the mosses, right? You walking through the forest, you see the green fluffy, that's like a big green fluffy thing, right? By the time you get to the liverworts, now we're talking about like this little flat green thallus. And yeah, you got some leafier ones, perella, but still pretty small. By the time you get to hornwort, remember the dime? Remember me showing you the dime with liverworts and hornworts and how small the gametophyte plant was? That's what I'm trying to hit home here. If you go this direction from the mosses down to the hornworts, the gametophyte plant size is still reducing over time, right? It's like still getting smaller and smaller. Now we're in the vascular plants. And today we learned about Lycopodium and Celgenella, right? For these, the gametophyte is no longer dominant. Now it is the sporophyte dominant. So when you see these green pictures here, you're looking at the sporophyte plant. You're not looking at the gametophyte plant. Here you were looking at the gametophyte plant. But here, these are sporophyte plants now. And we will see, we saw today, but we'll continue to see this continued decrease in gametophyte size. And this is one of these like core concepts of the course in terms of things that are important, is this reduction in gametophyte size in the evolutionary history of plants. Why is it important? Because basically, the spoiler alert, it's going to eventually lead to plants being able to make seeds, because you went from having homosporous spores with gametophyte plants that grew outside of the spore wall, right? Like here. And for Lycopodium too, but I'm not going to circle this because this is the sporophyte being pictured here. But for Lycopodium too, homosporous spores with exosporic gametophytes. Then you get to Celgenella. Now we have heterosporous spores and gametophyte plants that stay inside the spore, And this is a huge deal in terms of evolutionary history of plants, because ultimately this heterospory, look at the top of the slide, significance of heterospory. What's the significance? Ultimately, it will lead to seed shedding plants, seeds. So that's why it's super important. And as we learn about those seed-bearing plants, it'll become more clear. Okay. I think all that is left is to talk about this extinct Lycopod, Lepidodendron. The reason we're going to just touch on it briefly is because you will be able to see real-life fossil specimens, I think, in lab. And so I want you to know what you're looking at when you see them. So this is that picture we saw at the top of the class, right? In those Carboniferous forests where coal ultimately was made for us to use as fossil fuels. Lepidodendron was this super common tree. And it's weird to think, this is what's so cool about fossils and like learning about this plant evolution, because like Lycopodium, for example, and Celgenella, they're really small. Like you'll see some examples in lab, but they're very small things. They are not tree-like at all, but there were members of this phylum that were tree-like. They're now extinct, but it's just cool to think about a lycopod being a tree, because that doesn't exist anymore. So that's kind of cool. So Lepidodendron, this is this extinct lycopod tree. This could reach up to 100 feet tall and 6 feet wide, which is crazy to think about for lycopods, something that tall and wide. Just insane to me. It's almost like thinking of like a moss being that big. I know it's not the same thing because a moss is non-vascular, but it's how mind-boggling it is for me to think about. The top of the tree, so this here, what this is showing you, just so you know, this is a baby tree. This is like a teenager tree. And then this is like your mature 100-foot tall tree, 6-foot wide tree. When the tree is mature, at the top of the tree, you see a lot of equal dichotomy. So like look up here, right? A lot of equal dichotomy. And at the tips of all of the branches, remember they're not real branches yet, you have really massive strobili that are like this big, like huge. Not cones, as the book says. These are strobili. I will rant about that another day. So if you were to look closely at the stem, right? If you were to zoom in, say like right here, or zoom in like right here on the stem of this massive tree, this is what it would look like. And you would see all of these microphyls. They still were the same thing, even though they were bigger. Simple shape, one vein. They grew directly on the stem, just like in modern lycopods. But interestingly, it was a deciduous tree, meaning that the leaves would fall off every year, which is really interesting, particularly because these were growing in like a tropical environment, and that doesn't happen that often. It must have been a dry season thing or something. Anyways, when the leaves would fall off each year, it would leave behind a mark of where that leaf had been that kind of looks like a diamond. And if we were to zoom in on that diamond shape, we would see a few things. We would see like where the vascular system, or so you can call it a vascular bundle, but where the vein, basically, this is where that vein entered the leaf. Oh, and a silly ligule thing. So it's cool because we know these things by looking at actual fossil specimens, which show the same things. So, like, for example, here you go. Here's one of the diamonds, and that the vein of that leaf would have been right there. So I believe we should be able to see some fossil specimens in lab. Not for this lab, for future labs. We definitely have some fossils to show you, because Dr. Casper, who used to teach this course, is a paleobotanist. So like a extinct fossil hunter, like a fossil hunter of plants is what I think. Just to kind of hit some of these things home. So instead of looking at this, you can look at this. Here's the young trees that are growing, the baby trees. In the background here, you see those mature trees with the equal dichotomy. This is a close-up of a trunk. You can see the leaf scars, right, which is what we looked at here. And then giant strobili. Here's one giant strobilus hanging from the tips of the tree. So super cool to think about. Interestingly, this extinct lycopod was similar to Celgenella in that it was heterosporous, so it had megaspores and microspores and arranged, the leaves arranged in strobili, as I already said. And what was cool is the megasporophylls, or the leaves that would bear the megasporangia, actually served as little boats. So basically, here you go. This is a megasporophyll, and on it is a megasporangium. Inside of there would be megaspores. But basically, this little thing here, this megasporophyll, would serve as a little boat. So the megasporophyll would fall off, land on the water of the swamp, and the megasporangium would just still be on the little thing, and it would just kind of float by on this little boat. And then eventually what would happen is microspores would be released, and they would just like fall onto eventually this would eventually the megasporangium would open, exposing the megaspores, and then the microspores would fall from above as this leaf floated by on the water, which is super cool. Okay. Just so you have it for reference later. Okay, we did it. And perfect timing too. So that was a introduction to the lycopods, right? Lycopodium and Celgenella. Both of them are lycopods. Next time we're going to talk about horsetails. We'll talk a little bit more about the steel, which is not my most exciting topic, but it's important to cover. And then we will talk about the megaphyll, which we have not talked about yet. We've talked about megasporophyll, but we haven't talked about megaphyll yet. So we'll talk about that next time. Just so you know, so today, this class technically, so this lecture today is technically on the syllabus two lectures. I know. It's left over from when I used to spend a lot more time on the lycopods. So lecture 5 was the lecture of the introduction to the vascular plants that we did on Tuesday. Today, we technically covered lecture 6 and 7 today, even though it's really only the length of a single lecture. So there's an assignment for introduction to vascular plants and today's lecture, right? And that's due a week from today, the 17th before 10 a.m. like normal. So that should be available on Canvas now. Let me know if it's not. Cool. And with that, I hope you guys have a great weekend. Make sure you read your lab manual before going to lab. Ask me any questions about anything on the discussion or in an email, and I will talk to you guys later. Bye.